right. Hello, everyone. Welcome on into episode 2 of the Genetic Society of America on Twitch. Today, we have Dr. Jillian Freese here with us, who is an absolute legend. She is a published children's book author. She is a scuba diver. She is the co-chair of the Accessibility Subcommittee of the Early Career Leadership Program at the Genetic Society of America, and so much more. Dr. Freese, welcome on in.
Hi, everyone. Happy to be here today.
And we have a lot of folks in the chat already. We do have Jillian in the chat. We have Golganak. We have Smikes. We have a lovely as Jimmy B93, NMD96, and everyone else who's hanging out in that lurking land. Thank you all for being here with us today. Jillian is an amazing scientist, and her story hopefully will inspire more people to take up sciences. So, Jillian, let's start at the beginning. This is a standard opening question. What got you into science?
Oh man, that's a long story. Or maybe not too long of a story. My parents are both scientists, so I sort of grew up in a science household. But they were really great and encouraged me to explore all sorts of things. I went through phases where I wanted to be a writer and I wanted to be an artist, and it wasn't really until I got into high school I got my scuba certification for that was my birthday present one year, and uh, I got scuba certified, and I realized, wait, I can go out and go scuba diving, and that can be my job, and I can just study stuff that lives in the ocean, and that was just the coolest thing. And I never looked back from science after that point.
What kind of scientists were your parents?
My mom worked in
What brought you in? Was it seeing microbes? Was it seeing just the whole? Because as a kid, I always wanted to be a a aquatic marine biologist, or a scientist on the African savannah because of the big animals. What brought you into that realm?
I've always been really interested in how things interact with each other, and then how things interact with their environment. And growing up in San Diego, getting my scuba certification down here, a lot of that diving is in the kelp forest. And so the kelp forest is just sort of a perfect encapsulation of that sort of dynamic. There's a ton of organisms that are living in there. You've got, your sea stars and your urchins that are on the bottom, and you have the fish, and you have snails that live on the kelp, and then you have the kelp itself. And so really just sort of seeing the plethora of organisms sort of living in relative harmony. It is nature after all.
I like the relative abundance there on it. And a keystone species, says Chad as well, for the starfish.
Yeah, so that was really just sort of the impetus for me really diving deep on it. And I, had a ton of experiences while I was an undergrad, working on a variety of things that lived in the ocean. I sort of was, as long as it lived in salt water, I was willing to study it. I figured I could find something to love about every organism I was working on. And so I've worked on mussels and sand dollars, and I've worked on diatoms, and really sort of ran the gamut between things that are very micro and things that are very big.
So that's already leading to another question, right? How did you pick your undergrad institution? So that's always an interesting component of, were you looking for the research? Was it location? And then could you tell us about how you found a lab as an undergrad?
Yeah. So I did my undergrad at UC San Diego. It was basically in my backyard growing up here. There was a very small portion of it that was sort of family driven. Both of my parents went to UCSD. All three of us actually have the exact same degree. We all have a bachelor's in biology from UCSD.
That is really cool.
I added on a little bit. I got a minor in marine science while I was there. So mine's a little bit different, but overall we took the same path. UCSD is affiliated with the Scripps Institution of Oceanography, which is among the top tier oceanographic institutes in the world. And I knew that I wanted to be on a marine track, and that was the research I was interested in uh, doing. And so I sort of narrowed it down to some of the big oceanographic schools. It was UCSD, UC Santa Cruz has a really good program. Cal State Monterey Bay was also on that list. Um, and the strength of the research program at SIO um, really sort of sealed the deal for me in going to UCSD.
So that's all stemming from as a kid, right? Enjoying like scuba diving, and so that's really cool that you were able to jump into something that you were passionate of as a kid. Because a lot of us,
Mhm.
right? It's like no, you can't follow that anymore, and it's nice to hear that there are research opportunities for things that captivate you as a kid.
Yeah, definitely. It was, it was a combination of scuba diving and then the Planet Earth series and the Blue Planet series came out during my like junior and senior years in high school. And so I'm, watching that and I'm seeing the like crazy stuff that lives like in the deep ocean, and there's so many questions about how these things live and how everything interacts with each other. And I was just sold from that point on.
So
I was one of those weird kids who had life, who felt like they had life figured out and their career goals figured out at 14, while everyone else is still like, I don't know what I want to do. And I'm just like, this is what I'm going to get my PhD in as like a
so
high schooler.
you pretty much did, right? So
Yeah.
you're now a postdoctoral researcher.
Yep.
What so when you started in undergrad, you're already on that goal of okay, I'm getting that degree in that marine biology. Can
hmm
you just give us like what did you work on as an undergrad, and then we're gonna pivot to what your mindset was to getting that PhD afterwards.
Yeah.
Like what all like you said mussels and sand dollars and
Yeah.
diatoms and
So I worked in a couple different labs down at the Scripps Institution of Oceanography. I was a volunteer at the Birch Aquarium, which is one of the small. It's the aquarium affiliated with the Scripps Institution of Oceanography and UCSD, and I um, I was a volunteer there and was able to use the contacts that I had made as a volunteer there to get a position in a lab down at Scripps, which was sort of this like weird back road in. And so I actually had I was volunteering in a research lab before I started college in that summer. And so that was, probably the greatest. I was like, I don't care that I'm not getting a summer vacation that's like normal because this is the greatest thing I've ever done. Um, and so I worked with Lisa Levin's lab, and she works primarily on understanding the effects of climate change and ocean acidification on a variety of organisms, and focusing a lot on some deep sea stuff as well. So I was working on projects looking at, with another graduate student, looking at the effect of ocean acidification and increased carbon dioxide in the water um, on the development of urchin and mussel and sand dollar larvae, because those are all calcium carbonate based organisms. And so when you increase the amount of carbon dioxide in the water, it becomes more acidic, it eats away at those shells. And so we found that, significantly increased CO2 in the water does lead to like larval, like shell degradation. But there is a chance for them to still recover and sort of develop normally. It just takes them a lot longer.
so this would be in context of like global warming as well, right? Would be
Yeah.
the point of identifying that all these more things that can go wrong, right, with the
Yeah.
increase of carbon dioxide.
Yeah.
So you're an undergrad, you're volunteering in a lab you started before the semester even began, which is
Yeah.
that is so cool. And were you able to be there for the entirety of your undergrad in the same lab area?
So I worked in that lab for 2 years, and then I switched into other labs. I sort of bounced around a little bit. I took a microbiology course, a series of microbiology course, bacteriology, the whole gamut, and a genetics course. And that really sort of pushed me into wanting to get more sort of molecular and micro experience. And so that's where, some of the work with diatoms started coming in. We were looking at, and this is, tying it back to my desire to understand the relationships between organisms, where we're looking at the relationship between bacterial species in the water column and diatoms, because these diatoms can't make certain vitamin compounds. And so what they'll do is they'll excrete sugars, essentially, or other compounds, and then the bacteria will chemotax over to the diatoms, and then they have this nice little mutually beneficial thing where the diatoms give sugars to the bacteria, and the bacteria manufacture vitamins for the diatoms.
Really good. So it's still kind of that aquatic theme as well from the childhood love.
Yeah.
So you actually have a book on microorganisms.
Yes.
Did the idea for this book start during undergrad?
No, it was fully a PhD project.
Okay, we'll hold off on it, because I was like, there's a connection here that I'm excited about, like teasing apart. So you've gotten 2 labs under your belt, or a couple of labs, and you're identifying what you're interested in. You've gotten your undergrad degree, same as the parents, which is it's all 3 of all the same degrees, except you have a little bit of extra.
Yes.
How did you decide what your PhD was going to be in? Because you said, you kind of have already plotted out that, I wanted a PhD in this topic at the age of 14. How did that change when you were applying? And that, when you were applying, how were you influenced of where to go?
So when I was 14, it was the whole idea for what I wanted to do with my PhD was all ecology. It was, looking at how these things live and how they interact with each other, and it was just, very sort of, basic science as a 14-year-old. What you're exposed to in terms of like ecology and that sort of sense. I went down a pretty heavy molecular course load in my undergrad. I was really interested in genetics, and at that time, transcriptomics was starting to pick up. I am of the elder generation now. Transcriptomics was a thing. We weren't really doing proteomics at that point. If you were doing proteins, it was still like Western blots. And so I was really looking. It switched from ecology to molecular.
Was that a function of those courses that you took, or was that a function of the diatoms lab?
I think it was probably a mix of both. I mean, the courses were what got me into the diatom lab and that project. And then, that project was really interesting, and being able to do that sort of sequencing work to identify species and like identify genes that, were involved in this process was really important to me. And so it really sort of switched over to more of a molecular focus. I again was sort of, as long as it lived in the ocean, I could, find anything to be excited about. The big thing with picking a PhD program, especially for like the way that I did it, is like, because we didn't do rotations, I just went directly into my PhD lab. And so it is 99.9% of having a good PhD experience is having a good relationship with your advisor. And that 0.01% that's left over is being super interested in your project. Because if you don't get along with your lab mates, or if you don't feel supported by your PI or anything like that, then it's going to be miserable, even if you're working on your dream science project.
So just to set the stage for folks in the audience that might not know, usually what you have is when you join an institution, you'll do 3, 8 to 10 week rotations where you pick out, test the waters to go with the water theme of one lab. You do a second lab, and you do a third lab, and at the end, in the ideal world, each of those 3 have funding, and you pick which one you want to be part of. But so you went the alternate direction where you email ahead of time labs
Yeah.
that you might be interested in, and essentially you presumably did like a Zoom interview, right? And that's
Well,
how you.
this was pre-pandemic, so I did get flown out for in-person interviews.
So you do meet at least people in person. Then how do
Mhm.
you, for just members of the audience, like how do you determine in 15 minutes, basically, right, that you're going to spend the next 5 years in this
Yeah,
setting?
so I'm based in San Diego, went to UCSD, and they flew me out. I eventually decided to go to the University of Rhode Island. And for my interview there, they flew me out. I was out there for, I think, like 36 hours. Um, Took a red eye and spent the whole day in the lab with the people in the office, with the people that we shared lab space with, talking with all the other students, talking with other students in the program, talking with my future potential advisor, um, and just sort of hanging out with everyone and sort of getting a sense of how everyone worked together and what the projects were going to be like, and um, what that sort of dynamic was like. And so, it was 36 hours, so it was a little bit more than 15 minutes or 30 minutes that you might get on a Zoom call. But it, it was a really supportive group. Everyone got along, and the research questions that he was interested in asking were really interesting to me, even if the like primary goal of them was not necessarily like marine science.
So before we get into what the research was, we do have a question if you're familiar with the Mystic Aquarium in
I
Connecticut.
am. I am very familiar with the Mystic Aquarium. They actually do a Women in Science event every summer, or at least they have for the last, I don't know, probably close to a decade now, um, where they set up tables and booths all throughout the aquarium, and people and women in science, either in like research labs or in industry or in other aspects of science and STEM, will come to the aquarium. They'll set up their little booth and they'll interact with the aquarium visitors and teach them about all the research that they're doing. And we did that. Me and my friends did that every year. It was always a blast.
Oh, that's really so. What was your favorite part? Like, did you ever feel like? I know I've done some of those events at museums as well, and it almost feels like you're seeing yourself in the little kid who comes up and they're excited about marine science. Did you get that feeling as well? Like, what was your
Oh,
favorite part of it?
oh, every time. I mean, so I a little bit of a spoiler alert here. I worked on seaweed in my PhD, and so we would always go out. We'd collect a bunch of the like local seaweed species, and we'd take them in, and we'd have them in little like pools of in little bowls of water, so that the kids could touch them and learn about them. And we would have a microscope set up so that they could look at certain ones under the scope and see how the cells were arranged and that sort of stuff. And um, there's always an aspect of seeing yourself in the kids and seeing that sort of curiosity and excitement, but there's also just that curiosity and excitement that gets renewed in you from like getting to experience that sort of like secondhand from them. Having kids come up to the table and be like, ew, gross seaweed, and then like daring each other to touch it or to eat it and all that sort of stuff. And it's just like to see them go from like, oh, it's gross, it's slimy, to be like, oh wait, that's really cool, like look at this. Is always like a little bit of a heartwarming moment.
So we all were like brought microscopes and looked at the seaweed underneath the microscope.
Mhm. Yeah.
Cool. Was it?
Yeah, we'd have like a camera set up so we didn't have to worry about like focusing the microscope for each individual child.
Nice.
um, but yeah, no, if you could like put it on a screen and they could look through the scope and be like, and oh wow, like look at all the little cells and
I'll be honest with you. When I first heard about seaweed salad, which is very different from ocean seaweed, I thought it would look like ocean seaweed, and I had the same reaction as that little kid. I was like, "Oh no, I don't want to eat that." And yeah,
it's a little funny that I ended up doing my PhD on seaweed because growing up down here, going like diving in the kelp forest and stuff like that, kelp was slimy. Even if I was fascinated, with the kelp forest and everything in it, kelp was a little slimy. And then, I move out to Rhode Island to start my PhD, and a couple weeks later, I'm like elbow deep in buckets of seaweed, like just playing with it, like digging all around looking for stuff.
So, okay, that brings us to a whole bevy of questions. Number one, why seaweed? Like, what was the cool question? And then, did you have to go out to the ocean and do collections as well? Like, was there an element of still that ecology and the molecular? Like, I've got bevy of questions here in chat. Always make sure to ask your questions while we have Dr. Freese here. But yeah, what was the question? Why are we looking at seaweed?
So the lab that I joined was the lab of Chris Lane, and it was interesting being in that lab because most of the labs that I'd been in beforehand, everyone had been sort of working like on the same organism or the same type of organisms and was working on similar questions. And this lab, everyone worked on something completely different. So the whole lab focused on genome evolution and reduction in parasites, and then the relationships between parasitic, endosymbiotic, and symbiotic organisms. And so, like, I worked on parasitic red algae, but like one of my other grad students worked on apicomplexans, and other people are working on like other microbes and protists that are found in the guts of toads. So like we span the whole gamut of like weird organisms. So I worked on parasitic red algae, which are species of red algae that parasitize other red algae.
So are these like the red tide that we get, or is that different? Okay.
That's totally different. So red tide is caused by a photosynthetic organism called a dinoflagellate, and it's related to algae, but it's eaten algae a couple times. So these are multicellular, like multicellular algae, and they infect other multicellular algae. And it's so strange. There's about 120 of them that have been described, and only 3 of them are phylogenetically related. The rest of them are all independent evolutions of parasitism within this group.
Wow. Why? What is the evolutionary, like, there must be a selection pressure, right? What is that?
That's the question. That's the question. And it was just like, how did these things show up? How does the parasitism work? Because the other thing is that these parasites are really, evolutionarily speaking, young. And so we're catching them really early in the transition to parasitism. And so that allows us to sort of like see what genes are most important, or what loss of which genes lead to this sort of dependence on another organism. So the big thing is that these parasites have lost the ability to carry out photosynthesis. They still have a plastid, a chloroplast. They just don't have the genes necessary for photosynthesis anymore.
So that's what you mean by really new, because there's still, the machinery is mostly there. Like, that would do the photosynthesis, it's just that there's missing components already. So you might predict in, X number of generations you'll continue to lose, and presumably the chloroplast will be gone entirely one day as well. So what is it, the one you studied, how is its life cycle? Like, how does it reproduce?
So they make spores. So red algae are a little bit complicated. They go through a 3-phased life cycle. So I'll start them out as diploid organisms, and they make haploid spores. Those haploid spores are released and grow up as they go through meiosis. Haploid spores, those grow up as either male plants or female plants, and then those through mitosis make asexual spores. The males get released and then land on the female plant, where it fertilizes the haploid female spore, and then that will then be released and grow up into a diploid plant.
And are they then growing into other algae?
So that whole cycle happens the same for the parasites and happens the same for the host plant.
I see.
There's no difference between the two.
Wow.
They both go through that whole cycle.
Wow. So it's really sneaky on that front.
Yeah.
Smikes is wondering, with this work that you're doing in grad school, how are you served by the degree in both microbiology and versus like the marine biology lab that you'd had? And like, they sound really synergistic.
Yeah. So the sort of the micro and biology work that I had done really lent itself well to the molecular work that I was doing, and a lot of the manipulation that I did with the organism in the lab. So my project was I was really interested in trying to understand the development of the parasite, both morphologically, so what does it look like, and also molecularly, what genes are being expressed. Is it having to battle against like a host immune system sort of thing, in any way? And so my biology background really lent itself there really well. My marine biology sort of experience allowed me to sort of jump in pretty quickly in understanding the ecology and environment that the parasite found itself in. So this parasite lives on a red algae that lives in the intertidal zone, and so it senses through like dehydration and like increasing salinity levels, essentially, when the tide has gone out, and that's when it releases its spores. Because if it was at high tide, its spores would be released and then swept out to sea and swept everywhere else, whereas if it's released at low tide, those spores have a greater chance of landing and sticking onto an acceptable host.
So you were trying to figure out the sensor by which
that
it
was
would...
part of it, yeah.
That's really cool. Can you say, when can you say relatively recent? I think for the evolution of the parasite, like what's the time frame when we're talking about recent evolution?
We're looking at like a couple 100 million years.
100 million years or so, give or take. Nice. Okay,
Yeah.
so it's still, in order for it to lose more genetic information, such as the entire chloroplast, you'd probably double that length of time.
Oh, like more than. The thing is that the plastic, so the closest parasite relative to these is probably the apicomplexans. So like the causative agent of malaria is an apicomplexan. And they have an organelle that's called an apicoplast, which is essentially what used to be their plastid. It's just been super, super, super condensed over millennia, to where basically the only thing that it encodes is certain amino acid biosynthesis. And so, the parasite is getting stuff from the host. The red algal parasite does get stuff from its host, but it doesn't get everything, and so there is still some selection pressure for it to continue to maintain some.
And that's why it hasn't lost everything yet.
Yeah.
So maybe if it'll evolve into be a quote unquote better parasite, that's when you might see it lose more of its those fundamental genetic components.
Yeah.
So that is really cool work. What was the end question of your thesis, and what did you find with these parasites?
Yeah, so I was really interested in, like I said, understanding the development of the parasite. What does that look like? And so I did a bunch of sectioning. So I embedded the parasites at various stages in the plastic resin, and I sectioned them at a 5-micron thickness. So each section is 5 microns. I can't do the conversion, but it's really, really small. And I stained them with histology stains. So I used periodic acid and Schiff's reagent, which stains starches and sugars. And so that allowed me to both look at the structure of the parasite and how that changes over time, and also look at the starch concentrations, because like I said, these parasites have lost the ability to photosynthesize, and that is how plants make their food, is through photosynthesis. And so they've lost the ability to make glucose for themselves, and so that is the number one thing that they are dependent upon the host for. And so I was able to visually see the starch concentrations in the parasite. They start out pretty high because the spores are packed with starch to give them, enough fuel to get the infection going. So I'm able to see those starch concentrations decrease over the first couple weeks as the infection really gets established, and then watch the starch move from the uninfected neighboring cells to the infected into the infected cells, and then being pulled up into the parasite body.
So could you do live tracking as well, or is this something that had to be embedded because of just the tech?
This was something that had to be embedded. The cells themselves are fairly loosely packed together, and so it was difficult to sort of section them, like on a freezing microtome or something like that, because they would literally just sort of explode everywhere. So they really had to be embedded either in paraffin or in plastic resin.
So one question that we tend to get, especially with funding goes, is given how a lot of funding seems human-centric, for your grad student work, what kind of funding did the lab have? Was it like NSF funding? Was it for human-centric, or were you actually able to study it just because it's basic biology and it's cool? Which, right, all of these things should be studied, but we always have to justify ourselves nowadays. So, like, how did that work out?
Yeah, so my PhD was funded through an NSF grant. We were very fortunate to sort of have funding for I think there were two. I think there's now a third graduate student who's been on that grant, who's been funded by that money. The overarching goal of that grant was to sort of understand the phylogenetic relationships between these parasites and their host, and to do a bunch of genome sequencing, because we have, at the time that I was doing this, we had like 3 or 4 algal genomes, just period.
Wow.
Out of all of them, not even among the ones that and not even among the red algae. There was one macro algae, and there was one micro red algae, and then there was some green algae that had genomes. So that was a step forward, a step that we wanted to take forward, and I was just really grateful that the work that I wanted to do sort of fit into doing some of the genomics work, and also doing some of the relationship work.
Did you do a lot of, like, you did a lot of the imaging work? Did you also do a lot of sequencing during grad? So, like,
yes.
You were pulling in that molecular side of things as well. Did you also have to learn analyzing sequences on computers? Like, the training already sounds like you got a massive amount of stuff in. Like, what was that like?
So I had the morphology and histology side of the project, and then the other half of the project was a time course transcriptomics. So from week 1 so it takes 8 weeks or so for the parasite to go from spore to reproductive parasite. And so from week 1 to week 8, I had 8 different I had a time point every week of the development to see what was happening in the transcriptome, what was happening in the parasite. And so I have both parasite data, I have uninfected host data, and I have infected host data. And so I think the final total was about 63 million transcriptomic reads.
Ooh. And what did you find?
So there's it really sort of, the story sort of ties back to what you see morphologically as well. So we see a really big increase in sugar transporters and in endocytosis and exocytosis mechanisms that really sort of, show how that starch is being moved around the host.
That's really, really cool. We have some more questions for you. So on the phylogenetic front, do the parasite and the host have a common phylogenetic ancestor? And I guess, is that also true for a bunch of these organisms, right? Because you mentioned that it evolved a ton of times. Is there a close branch point, or like how does that work?
So the majority of these parasites are sister species to their host phylogenetically.
Wow.
Well, the parasite and the host are more closely related than the host is to other members of its genus.
Wow.
The most distantly related host parasite pairs are still found within the same family.
Wow. So
So,
what is the hypothesis there? Is it just a sub member of a given species underwent some crazy selective pressure and started parasitizing its original host species?
yeah, so remember how, so I was sort of describing the life cycle of these algae. So the like male haploid spore will land on the female plant and um, fuse with and fertilize the female spore. So that structure on the female plant is called a carposporophyte, and they make these diploid carpospores. And it's thought that there was some weird mutation in that spore that caused it to become reliant upon the host, because it's kind of like a pregnancy on a plant is sort of how I used to describe it. Because there's a single female spore and there's a single male spore, and it fertilizes, and then that goes through mitosis and they make thousands of identical copies. But all of this happens inside of this like round structure, and the plant, it's the haploid female plant, has these specialized nutritive cells that line this spore cavity essentially. And so if you imagine a spore that had some sort of weird mutation that wasn't able to make its own sugars, that it would try and tap into those nutritive cells that line that structure. And then now you have an organism that is reliant upon itself to keep it alive. And it's not too far-fetched to sort of see that additional mutations might happen there and sort of result in it developing into a parasite a little bit. It would also explain why these things are also highly host specific.
Definitely, and it's just wild of how many times it's happened. Like there must be something advantageous, right, about being a parasite on formerly yourself, almost, right? That's it's I'm echo Chad's sentiments. A little bit bonkers.
It is.
We have an ecology question as well. Do you know the distribution of the parasitic red algae, and has it changed with the increase in water temperature?
So they are found all over the place. There's a bunch in Australia and New Zealand area. There's a ton of them on the like west coast up in the Pacific Northwest of the United States. There's a couple species on the East Coast. There's a bunch over in like the Mediterranean and the Atlantic over on that side. Their range has expanded a little bit with respect to like climate change and global warming. The thing that's most important to remember with these parasites though is that they are reliant upon their host range. So if the host range expands, then the parasite range theoretically can expand. But the distribution and spread of these parasites is reliant upon the distribution and spread of the host, but also the ability of those spores to move to further and further away plants. So you end up with some of these species, like the one that I worked on, Coriacolax, that release their spores at low tide. They tend to have really patchy distributions. I mean, there was one site in all of Rhode Island that had these parasites, despite the fact that its host plant was present everywhere. And that's just because of the mechanism that it used to release its spores and ensure that it like would land on a right host. Other parasites release their spores underwater, and so they have a more broad distribution.
And that's what you were saying, actually. The reason that they're trying to get the different tides is because of how they're releasing it, and that's why when the tide is low versus when it's high, because it'll be going everywhere.
Yeah.
Very cool. I like when the mechanisms work back together with the hypothesis. For a quick reset, because we do have some new people coming on in, this is Dr. Jillian Freese, as in the Marvel DC supervillain, Dr. Freeze. We were chatting about earlier today. We were talking about her graduate work in red parasitic algae, and it turns out, Jillian, that there is also, you have a book entitled A is for Algae. This was during your graduate school career. We're going to pop the link into the chat because I think this is really, really cool. What was the idea here? Because you've talked about that you did, the science fairs for women in STEM.
Mm-hmm. Yeah.
And then all of a sudden, I see there's a children's book out there. Like, can you tell what was the thinking there while I share it to the audience?
Yeah. So the book is called A is for Algae. I had my comprehensive exams scheduled, and so I did my qualifying exam. My qualifying exam was to defend my dissertation proposal, and that passed. That was fine. And then you have your comprehensive exams, which were just this huge hurdle. It was 3 days of written questions that took all day. And then once you passed the written portion, then you had a 3-hour oral exam with all 4 members of my committee. And after spending, 6 very intense weeks studying for all of this, studying for the written portion and then studying again for the oral portion, I needed to de-stress a little bit. And so I started doodling, which is a thing that I do. And I never really had an intention of making a book. I was just doodling and playing around and enjoying not studying for a while. My cousin had my niece, my baby, her baby, maybe like 6 months to like a year, almost a year ahead of me doing all of this. And I am, other than my parents, I am the science one in the family. And so I knew that I wanted my nieces to grow up with a lot of science books. And there's just not a lot of like kid-friendly science books out there. And there aren't any about algae. And so I fully had the intention of just sort of taking all of my doodles, scanning them in, and then like just doing one of those like print books like you would for a photo album. It was just going to be a one-off just for them. I made the mistake of showing it to my mom and my aunt, and they were like, you need to get that published. Like, we're going to look into this for you. We're going to do everything we can. And so they were really the ones who sort of pushed it out and made it a thing in the world. It's gone so much further than I ever expected it to. It's in children's hands, and it's also it's a very popular gift for graduate students to give to other graduate students. I get a lot of messages being like, I got your book when I started my like graduate program, because I'm working on algae now.
Oh, that's beautiful.
And so like that's been really fun. It won the LH Tiffany Award, which is an award from the Phycological Society of America, which is phycology is the study of algae. So the big like algae society for the states gave it the L.H. Tiffany Award for expounding on the importance of algae in everyone's lives.
Wow, big congrats, Jillian. That's
Yeah.
awesome. Is it
Thank you.
like an alphabet book where it's like each letter of the alphabet is a different kind of algae?
It is. That is exactly how it works.
All right. Well, my little one will be learning the alphabet soon, so I'll need to pick this up.
Yeah, so each letter of the alphabet is a different type of algae. It switches between red, green, and brown, which are all of the different types of algae. There are macro algae, like giant kelp that, grow to meters and meters and meters long. And then there are teeny tiny little microalgae, like Volvox, that you couldn't see unless you had a microscope. And there's fun facts, and they've got little sizes on them, so you know how big they all are.
Ooh, I love me a scale bar. I love a
I
scale.
love it. Yeah.
Uh,
I tried to make it, I knew that I would probably end up reading it to my nieces, and so I wanted to make it, interesting for adults to look at as well.
did you include the algae that you worked on?
I did not.
Okay.
I did not. The algae that I worked on is not in there, mainly because it's not really pretty to draw.
Ah, okay, fair. And you did all the illustrations in this book, right?
I did all the writing and all the illustrations. And I worked with my cousin, who's a speech pathologist, to put together all of the pronunciations in the back, because I have the common name in there, but I also have a lot of Latin. And so if you aren't really sure on how to say something, there's a page in the back that will make sure that you can say it with confidence.
That is really cool. So this leads to like a whole other series of questions. Like, you're in the lab, you're this hardcore scientist, and then you also have this artistic side. And I think a fair number of younger scientists would argue that there is this intersection between art and science, right? Like, if you can be a good artist, you can explain your science in a very accessible way. How did you get up into doing these doodles, and then that, I know like the story of like for the niece and how you made the book, but like, have you pursued this further? Are you still like pushing this element of your life, like this really creative side, in addition to the science? Like, this is just such a cool fact about you.
Yeah, so after A is for Algae came out and it, was a thing, everyone in the family was like, well, what's the next book? And I was like, D is for dissertation. we're gonna finish the PhD first. And so since then, I've graduated, I've moved on to my postdoc, and so everyone's like, now I work on fungal pathogens, so everyone's like, F is for fungi. When's the next book? I think that the art really comes into like my science with my need to look at things. I'm a really bad microbiologist in that sense, because I like being able to see things and watch them. You can't always do that with micro. They're very tiny. So like when you're streaking out plates, especially at the beginning, I was like, I had a visible chunk on the end of my like loop. I was like, no, you don't need that much. But I think that that's really sort of where the artistic side comes in through, like all of the histology that I did in my PhD, like that's just plain, we used to call them watercolors, because you dilute them in water or ethanol, and then you stain your slides, and if you're not careful, it will stain you. So I think that's sort of where a lot of the artistic stuff has sort of bled into the science of it.
That's really cool. So the chat's already listing off the F for fungus, but we got the A and B already on in there.
Yeah.
You went from parasitic algae to parasitic fungus.
I did.
Is that okay? So I have to ask if it's still an ocean relative? Are we still on the ocean theme? And then, of course, how did you decide to make that kind of jump from like very direct marine organisms, like you've kind of been the love of most of your life so far,
Yeah.
to a parasitic fungus?
So at the end of my PhD, I sort of saw two different tracks ahead of me. There was one track where I stuck with the algae and I stayed with the marine realm. And then the other track was sticking with the parasite side of it and exploring that. And it was a combination of looking at job prospects and funding, because reality does have to sort of come into some of this a little bit. And also thinking about what part of it I was really interested in. Where were the questions coming from for me? And the algae side of things, it was interesting. I loved it. It's a really bonkers, weird system. And, if we could find more ways to support it or to tie it in with other like more relevant things, then like that was clearly a path that I would have gone down. But the parasite thing, I mean, it goes back to the kelp forest and me at 14. It's like I want to know how things are interacting with other things, because they can't talk. Like the fish are not talking and telling you about how they're interacting with the kelp and with the sea stars. You have to watch them and observe them and like see what's going on there. And so I really sort of was interested in the parasite story a lot more than I was the algae story. And I was sort of thinking at the end of my PhD, like a lot of people do, there's a couple different ways that you make a decision for what you want your postdoc work to be. You either stick with what you've done your PhD on, you stay on that same organism, you stay on a similar question. You can be really crazy and be like, I'm going to do an entirely new organism, I'm going to do an entirely new technique. Or you can do what I did, which was somewhere in the middle, which was a new organism, but the same techniques. So I'm still doing a lot of morphology work, I'm doing a lot of microscopy, and I'm doing a lot of transcriptomics, and now doing a lot of proteomics, and I've done some genomic sequencing here as well. But I'm doing it on a new organism, and that's the fungal pathogens.
So what is that organism?
So I primarily work on Histoplasma capsulatum.
Bless you.
Yeah, thank you. We also do some work on Candida auris here as well.
So why that one? Like, what is it just an interesting parasite? Does it also have like a human component? Like, why are you looking at that particular one? Like, what about its biology excites you?
So Histoplasma is a human pathogenic fungi. It causes respiratory disease, primarily. It's definitely a big deal in the human world. And it's a really interesting parasite in that it's unlike a lot of other fungal pathogens, because it's what's known as a dimorphic fungal pathogen. So they grow as multicellular filaments in the soil, and then if you inhale the asexual spores or fragments of the filaments, it will transition from a multicellular organism into a single-celled yeast inside your macrophages.
What?
Yeah.
So, Jillian, one more time, please. That's wild. That's
Yeah.
how,
So
why, what?
all of the important questions. Are you sure you're not a fungal biologist, Blaine?
I'm kind of like how you were saying, like, if it's something that is, you want to see how the world works, right?
Mm-hmm.
And that phenomena that you're telling me, going from multicellular to single cellular yeast within your macrophages, like, is it hijacking our immune system to achieve a new, like, part of its life cycle? Like,
So, no. So thermally dimorphic fungi grow as multicellular, hyphae or filaments in the environment, anywhere from like 20 to 27 degrees. And they transition to single-celled yeast or spherules within the host. There are 6 different types of thermally dimorphic fungi. And so histoplasma is found worldwide on every continent, including Antarctica. It's primarily found in bird and bat fecal material. And then, like I said, it's a respiratory pathogen. So when you inhale asexual spores and filament fragments, that's when the infection can develop. So you inhale these fungal fragments, and they are immediately recognized as foreign by your body. It's not self. And so your little immune system marches over, your macrophages make their way over and try and eat them up. And they are in the super acidic lysosomes, phagolysosomes within these macrophages. And that's where histoplasma starts getting weird, because they basically neutralize the acidic environment of the lysosome.
Wow.
And so they are able to divide within the macrophage.
So use almost the macrophage like a cocoon for development, like it's a little bag for them to just do what they need to do.
It's an invisibility cloak for them to hide from the rest of the immune system.
That macrophage would never be self-attacked because it's still carrying the signals that say I'm part of the system.
Yeah.
That is absolutely insanely cool.
It is.
We do have the question of why, but I think before that is because of this respiratory infection, is there comorbidity that's seen in patients as well, or is it so bad that this can wipe out a patient on its own?
So it depends on how much fungal material has been inhaled and the immune status of the person. Histoplasma is a primary fungal pathogen, so it's able to infect both immunocompetent and immunocompromised individuals, unlike many other fungal pathogens, which are opportunistic, like Candida albicans. So depending on your immune status, it could be more serious. It most commonly is limited to a pulmonary infection, with flu-like symptoms in the vast majority of people, but less commonly, and probably more commonly if you are immunocompromised, the infection is able to escape. Those yeast cells will burst out of the macrophage and disseminate to other organs and become chronic.
So that's the next step in its life cycle, is after it's reproduced in the macrophage, that cell bursts open. Then does it do it again? It's just a cycle.
Mm-hmm. As long as it's not being constrained by the immune system, then it will continue to divide within the host.
So do we know, or I guess maybe you're researching this, what are the factors that are driving this? Like what are the signaling molecules and the genes that we know that you're going from that multicellular state to single, and then do you go back to multicellular if you leave the host?
Yeah, so if I'm not going to use a human for this example, because it would be a little dark. But if you were a mouse out and you had been scratching around in the dirt and you were a mouse and you inhaled histoplasma and you got histoplasma and your body couldn't clear it for some reason and you died, then histoplasma would return to the soil and would return to the soil initially as yeast cells leaving the host, but would transition into filaments as the temperature dropped.
So it's a lot of it's based on that temperature dynamic.
It is entirely based on the temperature dynamics. So my postdoc research is mainly focused on understanding the relationship between morphology and virulence and how temperature is regulating that.
So what does that mean between morphology and virulence? Like, I know you were telling us that you're looking at under these players under a microscope. Is there a function of its shape and how toxic it's going to be to the system? And in that same vein, a question from chat is, what model organism do you use? It sounds like you use the actual fungus, the parasitic one. So
Yeah.
how do you keep yourself safe?
Yeah. So, I'm gonna start back a little bit. So, other fungal pathogens like Aspergillus, everyone's favorite, I know, don't change their morphology during infection. So, Aspergillus is filamentous when it's growing on your bread, and it's filamentous when it's growing in someone's lungs. Um, and so, really trying to sort of understand why um, histoplasma changes its morphology during infection and what role temperature plays. So, as a multicellular filament in the soil, histoplasma is not expressing virulence factors. But as a single celled yeast at 37 growing within a host, it's both a yeast and expressing virulence factors.
So if I were to able to turn down this is totally hypothetical. If I
Yeah.
could turn down my body temperature and inhale this fungus from the soil, nothing's going to
Mhm.
happen.
I'm not going to say nothing, um, just because I don't know how that would work.
But only like if
Uh,
my like this wouldn't happen, but if you kept your body temperature at that ambient temperature, it
yeah,
wouldn't form the virulence factors. So like it might be like growing in
yeah,
you, let's say, but it's not undergoing
yeah.
that same cycle versus do you see as you increase temperature changes in the structure that lead to like a line that's running along the same rate of like this is getting worse and worse for you?
It's not quite that clear. There's so like at 25 degrees, it's definitely a filament, and at 37 degrees, it's definitely a yeast. And like 30, 32 degrees, 28 degrees, like in there, there's a sort of a like range where some of them are still filaments and some of them are yeast. And so you've got sort of a mixed population more at that temperature. Um, if you were able to lower your body temperature to 25 and have all of your immune system and everything still work perfectly, the fungus would your immune system would probably be able to clear the fungus. Um, One of the really weird things that histoplasma does in the infection process is that it changes the makeup of its cell wall. So it goes
As it's
from,
changing the temperature.
well, so you're, I think it's alpha 1 4 glucan, is the like primary cell wall protein at 25. And in yeast cells, that alpha layer is covered with beta glucan because it's less recognized as a pathogen associated molecular pattern.
So again, it's
And
its
immune system.
sneakiness.
It's a little bit sneaky.
Yeah, that is. So it's circumventing the immune system instead of directly attacking it.
Hmm.
It it's such a cool organism.
It is.
How do you stay safe? Like when you're
So,
working with these samples, like how did Dr. Freeze not get this infection?
so there's a couple different like things at play here. The first thing is always important is PPE, just personal protective gear in the lab. I work in a BSL-2 and a BSL-3 environment, so that comes with additional safety mechanisms and
So
protocols in
just
place.
to reset to folks who maybe don't know what that means, that's like higher tiers of security based on like that biosafety level, and the higher the number, the higher restrictions that you have, right?
Yeah. So in BSL-3, sometimes you wear a respirator. You wear like a bunny suit essentially. You're all geared up, and you work, with other people so that you have a buddy system. Um, the other sort of aspect of keeping people safe is that we have attenuated virulent strains. So one of the strains that we work with most often, we have engineered so that it is missing the URA5 gene that um, encodes one of the products necessary to make uracil within the cell. And so you have to add supplemental uracil to your media in order for it to grow. But if I were to inhale spores from that strain, or if I were to, get really thirsty in the lab and drink my flask of yeast for some reason, um, it wouldn't be able to survive in my body because my body would not be a source of exogenous uracil.
Do you find, are the results at all changed as a way of working with this attenuated strain, or do you find that removal of this gene really doesn't influence the factors that you're studying, so you can still like infer the behavior of the fungus with or without this gene, like what you'd
Yeah,
see
so
in the wild?
it depends a lot on which aspect you're looking at. So if you're trying to study nucleotide biosynthesis, obviously impairing the pathway for generating uracil is going to be a problem. But that's not ever been really a focus of my research. So I haven't experienced any sort of negative effects on the targets that I'm looking at. But we have the ability to, and we have done comparative experiments between the wild type strain and the Ura5, the uracil attenuated strain, and not seen any like super significant differences for our work.
Cool. And that's a nice safety feature as well, that you feel like you can work with them and not have to worry about any other off-target effects on you. So
Yeah.
what does a normal day look like for you? Like you said, you mentioned bunny suit, right? That you have to get, like, how often is that something that you have to do? Like, for a fungal geneticist such as yourself, like, what does your day-to-day work look like?
It really depends on what's going on. So I tend to go through booms and busts of experiments where I will be really busy and I'll be running, a dozen, what feels like a dozen experiments all at once. It's really more like two, maybe three. Um, but they're running together, and I'm juggling a bunch of different aspects of the work that way. Um, and then other days it's really heavy computer analysis. Like I said, we're doing a lot of sequencing work, so a lot of transcriptomics, a lot of proteomics. We finished up the genome sequence for histoplasmas.
Oh, cool. So
Uh,
now it's easier to do the transcriptome work.
yeah, so so that paper came out in early 2021 with some collaborators up at UC San Francisco and with the Broad Institute at MIT, um, doing the full genomes for a bunch of different strains of histoplasma. But prior to that, the histoplasma genome was like 206 contigs, so pretty good, but not great. But again, like, you have to sort of think about like where people are coming from with their experience, because that colors it. So I'm coming from an algae world where I didn't have a single genome and everything was done de novo, and so it's like, oh, 200 contigs, this is great, I can work with this. And then there's other people who come at it from like a bacterial side or from a human research side, and they're like, what do you mean you don't have the complete genome telomere to telomere yet? What do you mean there's unannotated genes? So
Yeah, I'm working with ants. I feel you on the unedited genes. It's like, what is this? I don't
yeah,
know. What do you mean you don't know?
yeah, like 56% of the histoplasma genome doesn't have an annotation. It's like hypothetical protein.
So do you ever find yourself, like when you said you're doing the transcriptome work to look at the RNA, you're looking at the proteomics to look at the proteins.
Yeah.
Are you finding interesting results that are like really highly or lowly expressed that are unknown? Like you don't know what
Yeah.
their function are, and then if you do, like what do you do? Like what's your next step there?
Yeah. So my research, like I said, I'm really interested in understanding the temperature aspect and how temperature is regulating things. And so one of the most well-studied responses to temperature changes is, of course, the heat shock response. So my research focuses on understanding the role of heat shock protein 90, and what role that plays in regulating the cellular response to temperature changes. So HSP90 is the most abundant protein in your cells. It's 1 to 2% of total protein in unstressed cells, and when they're under heat shock conditions, that can jump from anywhere from 4 to 10% of all cellular protein is HSP90. And it interacts with a huge number of genes. So in the model yeast Saccharomyces, HSP90 interacts with up to 10% of all proteins.
Wow.
And so this protein assists other proteins to fold properly, it stabilizes them against heat stress, and aids against protein degradation. So I like to describe it as like a Tupperware, because it opens and closes, and it is assisting all these other proteins that are termed client proteins. And so the structure opens, it's like taking the lid off the Tupperware. The protein goes inside the Tupperware, the lid closes, the protein closes, and then that Tupperware, you can carry it around the cell to where it needs to go, and you can protect it from all the heat stress or the cold stress or whatever's going on in the cell, and you can prevent that protein from being totally degraded. And if it turns out that protein's misfolded or something, you can refold it, or if it's not folded correctly at all, you just put a little tag on it and send it out in the world, and it gets picked up and degraded and recycled back into the cell.
Gotcha. So is this also an example, like from earlier in chat, where folks were asking the techniques you learned previously, like in graduate school, you're better able, now you're utilizing in your postdoc, especially when looking at those genomes. Is it any real different that you're working with a different organism, or are they the same principles that you used in graduate school, now you're just applying that in your postdoctoral work?
Yeah, it's a lot of the same techniques and a lot of the same methods. Like I said, I have a genome now, so that makes my life a lot easier, for sure. But it's a lot of the same game still. It's molecular work and genomics and identifying genes and all that stuff is still very dependent upon how much has been done in the field. How many resources do you have? What's in the database? Do you have a lot of stuff in the database, or are you the first one doing that sort of experiment? And with my PhD work, like I was the first one to do a time series molecular study on the parasite, and so it's like I didn't have a lot of references in a database to sort of compare my data to. I have a lot more now, which makes my job easier, but there's still a lot that's unknown and unannotated. And so then you go back and you're like, well, there's a lot of other fungi that have been sequenced. You're looking for homologous sequences. You're looking for, you're putting those proteins into things like Uniprot, and looking for conserved regions, or, like, oh, this is an alpha helix, and that sort of structural similarity.
How do you find working on both sides of the bench? So this is something that I know a lot of our peers have difficulty with. I know I did, of working on the computer, like the bioinformatics side of things, and then at the actual bench doing like the imaging and these, like you're doing both worlds.
Yeah.
Was that a difficult set of skills to learn, or was it just like what like how did you end up feeling proficient in both?
Yeah, I had a lot of experience with microscopy simply due to like the work that I was doing in my undergrad, and like just the lab courses I was taking in my undergrad. I sort of had a good basis for that ahead of time. I also just had really good mentors, and people who had done that experience, who had done that stuff before, and felt comfortable teaching me about it. Is just also super important. So like the lab side of things, I felt like I had fairly under control, or at least I had some confidence in myself in. The molecular side of things, especially all of the computational work, I didn't take a biostats class in undergrad. They were not required. I did not take a computer science class in undergrad. It was not required. I would recommend that people in biology that want to do research take both of those things now. So I took one course my first semester in graduate school. The half the semester was learning Python, and the other half of the semester was learning R. And I have been entirely self-taught in all the bioinformatics analysis after that point in time.
Wow, that is not easy. That is like
Yeah.
0%. Because I know I think back on my first ecology course, and they
Hmm.
told us to make a box whisker plot. I thought it referred to a cat. Full disclosure, no idea what they were talking about.
Yeah.
And that's just really impressive that you're able to then both know both languages under your belt, and then you use both for the sequencing and I guess proteome analysis as well, right?
Yeah. Yeah. I like working in R better. I think that also sort of comes back to the artistic side of things. R has better figures in my
Is that we have
in my
in chat
opinion.
a GG plot for the win?
Yep,
Is
that is a GG plot is pretty great.
nice.
Definitely not gonna say anything bad about GG plot. Everything for me. But it's a different sort of challenge and sort of like picking all of that up. I think that the most useful thing that that class that I took taught me was a lot of the like terminology, because even people who have done a lot of programming and stuff before, it's about knowing how to ask Google your question, because you can know what the problem is, or you can sort of like figure out um, what it is that you want to do. But if you don't know exactly like what you need, like do you need to set up a dictionary in Python, or do you need to like set the aesthetics in R? And if you don't know how to ask Google, or yeah, now Chat GPT, how to write your code for you, like you're not going to get an answer, and you're going to get really frustrated. And so I think that sort of learning how to ask the question correctly was the most useful thing I got out of that class.
Or how to search Stack Overflow. That from the chat it brings me back
What?
as well.
Yes.
We did have a question about the organism of why did it take someone so long to get a good sequence of the fungus that you're working with? Like, why is it a question of price? Because I know that comes down with time and it's easier to
Mhm.
sequence a whole genome. Is it just that you were the scientist, the pioneer interest in it? Like, why did it take so long of something that we're talking about how important this would be to understand and how cool the system is, not just from a
Yeah.
fungal perspective but basic biology. Like, why did it take so long?
Yeah. So histoplasma has a really strange genome. I mean, a lot of fungi do. Um, as a group, they have the greatest fungal genomic diversity across all the other organisms. Like just within Aspergillus, genome diversity among the sequenced species, so not even all of the species, just the ones that we've sequenced so far, um, is as diverse as that found across all vertebrates. So very
Wow.
close relatives, Aspergillus fumigatus and Aspergillus fisherinus, are as divergent as humans and mice. And they're all within the same
That's
genus.
what I was. They're in the same genus. Like, what?
Yeah.
So what is driving that? Like, why are
So
they so different?
it depends on where they're found. It really comes down to where they're found in the environment. So like Aspergillus fumigatus is a common human fungal pathogen, and other species are, there to eat your bread or to digest leaf matter. And so there's entirely different sort of metabolic capabilities, but they have the same sort of backbone in terms of, the core housekeeping genes are still really highly conserved and similar. That's just a really small set of their genome, because they have evolved these really sort of diverse metabolic capabilities.
And so, for the question in chat, is the reason that they're in the same genus is because that core of set of housekeeping is so fundamentally similar.
Yeah, that's exactly it. That's those genes that we use to identify organisms, your small ribosome sequences, your large ribosome sequences, your ITS sequences, those are all still really fairly conserved, or at
Wow.
least conserved enough to put them all in the same genus.
It's cool that we've had two different phylogenetic discussions today, right? Like with your
Yeah,
undergrad work, it's they're near identical, right?
yeah,
They're just like the parasite is like so close to the host, and it's
hmm,
just like they're just side by side by side. And now
yeah,
fast forward to your like the postdoc work and other aspirate fungus, we're speaking about
hmm,
huge amount of divergence. So it just speaks
yeah.
to a lot of the cool science that can be done with fungi.
Yeah, so histoplasma's genome has these really large repeat regions, and depending on the strain and how they're grown and stuff like that, those repeat regions will shrink or expand, which is one of the reasons why it took so long to get the genome from 206 contigs down to the 12, to the like 6 or 7 chromosomes. And so it was really a combination of Illumina sequencing, so short read sequencing, and a lot of MinION Oxford Nanopore technology, the long read sequencing, that allowed us to sort of combine the two together and sort of span some of those repeat regions to close those gaps.
Cool. We have a question that I think you're well expertise to answer. So it seems like 75 years ago, taxonomy was looked down upon by real scientists. Has that
Mhm.
changed in your opinion with the onset of genome sequencing?
Um, it depends on which field you're in, I think, because genomics has made taxonomy easier for some people, and it has made taxonomy nearly impossible for others. So, taxonomy for the algal field is great. Um, you go out, you grind up your seaweed, you sequence it, and you find out what it is. And there's a huge backlog of things that have been sequenced that need to be redescribed and given new names, and like reorganized in the phylogenetic tree. And it's literally, there's just too much to do for the number of people in the field. Um, and it's also complicated. Algae and fungi were both initially described by Linnaeus, and so there are still species that have Linnaeus as the naming authority on the species, which is both really cool and kind of crazy, that name has stuck around for so long. But it also means that there are a lot of rules about naming and renaming these species, and so in order to have it, there's a whole book on how to name algae and fungi and plants, so that they are considered valid by the field. So there's a lot of work to do for like the algae world, and I'm sure for fungi as well. Um, but if you work in bacteriology, if you're working with Vibrio, you know, taxonomy becomes a nightmare because there are so many little changes that happen, and it's like, how do you define where the cutoff is going to be for when something is too divergent to belong to that species anymore?
Yeah, as a member of chat says, the answer to what a species is, changes over time and point of view constantly. Same
Yeah.
thing with what is alive and what isn't, right? Like, is a virus
Yeah.
alive because it has some features, especially with parasitism, like you're studying.
Mhm.
Those are
Yeah.
some arguments as to why viruses aren't alive because they don't
Yeah.
make X, Y, and Z. And then you can come in with like, look at my parasites. You'd say
Yeah.
that they were alive, but they also don't make those, and
Yeah.
it's always a cool way to see those walls broken down in these definitions, and it's just fun to find out how
Yeah.
that works.
We, as scientists, we love putting things in categories and giving things names. But it's also important that we recognize that those categories and names are arbitrary.
it, uh, so, question: there are 250,000 species of extant beetles. Do you have
Mhm.
an estimate as to how many species of extant fungi there might be?
Um, I think we're in the millions.
Millions. Wow. There you go, Smikes.
The I'm gonna sort of okay. So quick Google here, because I had to double check myself. So 148,000 species have been described of fungi, but the like true biodiversity is estimated to be between 2 and 4 million.
Wow. So we've got a long way to go.
There's a long way to go.
Wow. So speaking of wow, in addition to your postdoc work, you do a bunch of other stuff as well.
Yeah.
One of which you are at the Genetic Society of America as the Early Career Leadership Program co-chair of the Accessibility Subcommittee, which is a mouthful that I've just like spewed out. Can you tell us a little bit about what drew you to be an advocate for accessibility in science? What that means to you? Like, what is like? First of all, I guess what is it? Second of all, why do we need an advocate for it? And I think these are all important. You and I both know the answers to this, but I think it's a great stage of kind of setting forth why these are important factors, and then what drew you to the program at the Genetic Society of America, and what are you doing? And you can highlight some of the cool achievements that I know you've done in the program.
Yeah. So in the U.S., disability is defined as a person who has a physical or mental impairment that substantially inhibits one or more of their major life activities. It's important to note that that is a legal definition of disability and not like a medical definition of disability, but that's the one that we use for all of our metrics. NSF and NIH do surveys to sort of assess what the diversity and the people in the field look like and what they are doing. And so, that's, in the US, about 27% identify as disabled now. That's about 1 in 4 people. That disability may be visible or maybe invisible. You might not see it. And it's the only minority group that you can join at any time in your life. And so, there's a large population of people who identify as disabled or who have a disability. But only 10% of STEM undergrads identify as disabled, 6% of graduate students, and less than 2% of postdoctoral students. So you can only imagine how many fewer percentages there are of PIs that are submitting grants and what the grant funding situation looks like for those individuals. Yes, this does include mental illness. That definition does include mental illness. And so, diversity, equity, and inclusion efforts have sort of, really sort of cropped up and been a big push in the last, decade or so now. But accommodations for disabled scientists or inclusion of disabled scientists in those or just disabled people in those conversations are either not a priority or have not been considered at all. And so like that's, a gap that really needs to sort of be addressed. I was diagnosed with Ehlers-Danlos syndrome. It's a joint condition where my collagen doesn't quite function the way that it's supposed to. And so I had to deal with that through graduate school, and I went to the accommodations office, and like many other disabled graduate students and even disabled undergraduate students, the resources that are available in those situations are not necessarily going to benefit everyone. And there's a lot of hurdles that you have to overcome to use those offices. You often have to have a diagnosis. You have to have essentially a doctor's note in order to actually have access to the accommodations that office provides. And they're primarily focused on accommodations for classroom activities, so teaching and lecturing and, test taking sort of things. Accommodations for field work, accommodations for, lab work and being a teaching assistant, like those were not things that office that those people are prepared to handle. And so having an accessibility subcommittee within GSA, having accessibility groups on university campuses and businesses, um, are, essential for making sure that everyone has access to the things that they need in order to succeed.
So were you, with respect, like in graduate school, were you able to get anything in terms of support for, because you were saying you were doing field work, right? You were collecting the
Yeah.
red algae, and
Yeah.
then of course that's like we have members in chat too who are also in labs saying that they, like, there could be the chair that you're using
Hmm.
pipetting,
Yeah.
right, can be something that you need accessibility to. I know I have multiple shoulder injuries, and I found pipetting really difficult because of how
Yeah.
much pain it would get in, and the reaction was like, "Well, knock it off,"
Hmm.
right? And so,
Yeah.
how do you deal? Because you're finding, like, because the numbers you gave, one could very easily argue they're underreported, right? There's no way that's the real number, and so it's like you're fighting an uphill battle. So,
Hmm.
I guess, how did you become passionate enough about it to take on that uphill battle?
Yeah. So, the experience that disabled students have in graduate student labs, in undergraduate labs, as postdocs and all that, is entirely dependent upon the support provided by their program, by their advisor, by mentors, by their other graduate students. And so it's all very individual experiences. So I had a lot of support from, other members of my lab, my friends, and my PI. And so, like, we would find accommodations, or we would find, we could try and find workarounds for things that were, problematic for me in any way. Um, and it was really sort of, I was also in graduate school. I was part of, um, the I helped found the first chapter for the Society for Women of Marine Science, which is now a nonprofit 501(c)(3) organization with like members and chapters worldwide.
Wow.
And the goal of it is, the goal of SWIMS is to bring together marine scientists of all career levels to discuss the diverse experiences of women in marine science, celebrate the research done by women in the field, and promote the visibility of them in the community. Um, and it was at these, we do these day-long symposiums, um, where, we talk about career opportunities, or they're focused on like mentoring and, that sort of thing. And it was at these symposia that, we started talking about, we had this group called Seasick. And it was, we're into the puns. But it was talking about, mental health and physical health and well-being and disability in the marine sciences, because a lot of marine science research can be really physical. Like, these are women who are going out on research vessels, or are going into subs and doing deep sea work, and like, the accommodations for that are not going to be something that the university provides. It's stuff that you crowdsource. um, and so it was, for me and for a lot of the other members of the accessibility subcommittee, it's about doing what we can to make sure, as much as we can, that there's sort of a guideline and a structure for making everything as accessible and available as possible.
So can you talk about some of the achievements that you've led on the subcommittee? I know I'm gonna, I'm gonna brag on Dr. Freeze here, but there is, from the Otter AI at the conferences to some of the seminars and tools that you've put forth. If there's anything you want to highlight, because it's a, I still feel like, again, it's an uphill battle. It's underreported. There is an inherent stigma against it in many cultures. Like I said, when I was trying to
Yeah.
pipette things, lab mates would be like, knock it off, you're being silly, right? Like, so can we just highlight and celebrate your achievements on that committee for a moment?
Yeah, so the Accessibility Committee is a big proponent of universal design, which is the idea that an environment or thing, product, whatever it is that you're doing, can be accessed, understood, and used to the greatest extent by all people, regardless of age, size, ability, or disability. Um, and we really sort of like to, say that if an environment is accessible, usable, convenient, enjoyable to use, everyone benefits. You know, we put captions on things for people who are hard of hearing or who are deaf. But I watch Netflix with the captions on all the time. They're beneficial for everyone. Um, so, those sorts of things are really sort of driving mechanisms for us. We've implemented the use of Otter AI in all of our meetings and seminars and workshops.
And that's
There's been a
a
lot of,
transcript service, right?
yeah,
Yeah.
that's the transcript service. There's been a lot of accommodations and guidelines that we've been able to put in for GSA conferences. No one really wants to stand next to their poster for 2 to 3 hours, but we do it anyway. But we've gotten chairs put next to everyone's poster so that you can sit, or if you don't want to sit, you at least can put your stuff somewhere other than the floor. Um, you know, so that's sort of those things. Some guidelines as far as color choices and font choices and font sizes on posters and slide presentations. And then, of course, we have the accessibility and STEM workshop series that we've had going on. We've been able to talk about accessibility in teaching and accessibility in the lab and in the field. And then we've been developing a lot of other tools and resources that are, giving people more guidelines and more opportunities to make events, presentations, and workspaces accessible to all.
So I did a shameless plug for the YouTube page where you can actually see some of Dr. Freese's seminars that she's helped organize, lead, and moderate. Can you tell us about like one of your favorite seminars or like bits of information that you learned that you didn't know until after the seminar that was a very worthwhile thing that everyone should know about?
Yeah, I'd probably go back to our first seminar, which was on accessibility and teaching. Um, there are a lot of factors that you have to sort of take into account there because whether you're the lecturer or the student, the accommodations need to work for you, but also for everyone else. And so there are instances, and I like hadn't really considered this because I'd never run into it, but there are instances where you will have potentially multiple students in a classroom that have accommodation needs, but those accommodations may run counter to one another. Like you may need someone who is hard of hearing and needs a translator and has someone who's signing ASL in the class, but then you can have someone else who has focus issues or something like that, where the ASL interpreter is really distracting. And so trying to find a balance and sort of like take multiple accommodation needs and find a way to make them all work was a scenario that I had not considered, but that I'm glad that other people are.
Yeah, because that seems like it would be a lot of issues would arise and like the best way of taking part in it. Yeah, and if folks are interested, the video is uploaded onto the YouTube page.
With the transcript.
Yes, with the transcript. So actually, what's really cool that Jillian mentioned is using the Otter AI service to have transcripts available, and the subcommittee always goes through and makes sure that the transcripts are accurate. They correct things that the service doesn't get and upload them to YouTube with the appropriate transcription. So again, it's making accessibility much more of the norm and not a extra accoutrement, as it were.
Yeah.
What, Jillian, are you most excited about? You mentioned some of the new tools that you're thinking about developing. Like, what is on the projects of the committee's horizon that you're really excited about?
Yeah, so we've been working on putting together a toolbox, and it's broken down into essentially it's like whether you have the disability or whether you know someone with the disability. And then it's broken down into whether you're doing a poster presentation, whether you're doing a slide presentation, are you making other materials for promotion, are you teaching, are you giving a talk? Um, in like are you planning an event, like every scenario that you might run into as a scientist working in STEM. We've got multiple resources for checking to see if your thing fits with accessibility guidelines, um, and also giving you tools to start designing them more, to design everything with accessibility in mind from the very beginning, because that makes it easier, because then you don't have to go back and do it again. um, and also just putting together resources for uh, accommodations um, that you might not think about. um, so like I have Ehlers-Danlos syndrome, so like my accommodations are mostly going to be involving physical things, it's using scissors to cut parafilm, or that sort of thing. And like having additional grips on like a pipette periodically, if my joints are acting up, or having the right sort of chair in the office. But if you're hard of hearing, or if you are blind, or have poor vision, the accommodations that you're going to need in an office space or in a lab space or in a field space are very different than the ones that I might need. And so we're trying to put together a resource that will give multiple um, sort of starting points for people to think about, oh, that might be really beneficial to me, and I wouldn't have thought about that.
That's really cool. And one thing that y'all do that I want to highlight as well, that whenever there's proposals in the program of a new something coming forward, you and your subcommittee always review it to make sure that that it is accessible to all. And that's a really nice filter that actually you led to put in place to make sure that there's always accessibility on not just your subcommittee, but all of the subcommittees in the early career leadership program, which is, again, I think it's so cool that we have something like that.
Yeah. I will direct everyone to askjan.org. They have an A to Z of disabilities and accommodations. It's a huge resource. I'll throw the in the chat. But it's like if you have a condition, or if you start working with someone who has a condition, or who has a disability, it's a great place to sort of start, and look for what um, uh, uh, what accommodations might be beneficial for them in the workplace.
Cool. Thank you. So, Jillian, as we begin to wrap up, one question that we always like to ask is, what is the end goal for Dr. Freese? What is your dream? Like, where would you like to end up one day? What is the overarching goal, and how does one get to some kind of goal like that?
Yeah, I mean, you're supposed to always have a 5-year plan. Let's just say that. It's the future is still a little murky for me. I want to stay to finally kill Batman. Yes, the Penguin will take him out eventually. I'm still working on my costume though. If anyone's got any great ideas, Halloween is coming up. Um, but one of the like big things for me is that I love research, and I love being involved in research. Directing my own research group is always a dream. I think that if moving forward in that space, I'd really love to move into a translational sort of research space. A lot of my background and what I've done in the past and what I'm doing currently is a lot of basic research, trying to understand how do these organisms work, what's going on, and sort of taking what I'm finding um, take what I'm finding in terms of target genes for new drugs or other new small molecules that might be more effective at killing or preventing these infections, and being able to take those up to the stage where they're ready for a clinical trial. I think would be a really rewarding sort of career. And at this point in time, that's I'd love to be a PI. I'd love to be the one, fully directing that. But there's also a lot of opportunities in government and industry research to sort of do that same sort of work.
Do you plan on continuing the book series?
I'm gonna say yes. I'm never gonna close the door on it. But it's harder with fungi. They're microscopic. It's a lot of yeast. It's a lot of round yeast, and it's a lot of filaments. It's I'm trying to find some more fun things to draw.
So
Always.
what do you do for fun now? In addition to
Um,
still just the art, right? Like what's some of the other things?
in addition to the art and that sort of stuff, I do a lot of hiking. I'm in San Diego, so there's a lot of outdoorsy fun stuff to do around here. So it's a lot of hiking. We had a great snow season, so lots of skiing this year. Um, yeah, I do love scuba diving. I do not surf anymore. Um, there's some. It's always fun to go dive in the kelp forests and stuff around here, of course. But there's also some pretty cool wreck dives around here. They've sunk some old like oil towers and another ship off the coast as artificial reefs, and so
So
they cut big holes in them so you can like go swim through them all. It's very fun.
no sharks, right? I always just assume that it's like a scene from Finding Nemo where you have, Bruce the Great White come up and
We have sharks. Um, La Jolla has the leopard sharks that come into the cove every year, and they're in like 3 feet of water. But the leopard sharks mostly eat shellfish,
okay.
so they're not going after you or your toes. So when the water is clear, it's a lot of fun to sort of just sort of wade out, and there's all these pregnant female sharks swimming around your feet. It's very cool.
That does sound really cool. And now, no, I'm not arguing the coolness of sharks. I love sharks. I just had this vision of the sunken ship, and there's always like the bull shark, because in Florida,
Yeah.
that's what would be swimming around.
Yeah.
Being
That
a
is
little
true,
bit more
yeah.
on the careful side. So it's cool that you're still able to go. Like it kind of brings us full circle, right? The
Mhm.
initial love of science for
Yeah.
you was in that scuba diving,
Yeah.
and you've come back to that even during your postdoctoral research, be able
Yeah.
to get back into scuba diving
Hmm.
and still do that exploration. Do you find that
Yeah.
do you still have as much love of scuba diving as when you did as when a kid?
Oh yeah, definitely. I mean, it's fun scuba diving now because it's like more of a hobby than it is a job, right? So it's really it's just pure fun to go out now. It's not like, oh, I need to like go and collect these samples or whatever. Um, but it's also, I know so much more about it now, and so I'm the nerd that's out there being like, hey, did you see this species? Or like, I'll be grabbing someone's fin and tugging them and being like, hey, look at the sea cucumber, and they'll be like, but there's a shark over here.
Do you find
Look
yourself
at the stingrays, like.
do you collect do you like I know you don't have to collect any samples when you're out there, but do you find yourself like I'm just thinking like, gosh, it would be really cool to put this under a microscope?
Oh yeah, all the time, all the time. I'd love to. Everything should go underneath a microscope.
I love that mindset. I'm going down to the beach next week, and I'm going to be bringing vials just to bring back stuff to put under a microscope. I don't even
Yeah.
work on it, but it would just be cool to see
Oh yeah, throw some sand in there.
some
There's all
sand.
sorts. Yeah, there's all sorts of fun stuff in sand.
Okay, very cool. And yeah, we can always next time if we have you back, we can if you send us some samples, we'll happily throw them under the microscope while you're live here, and you can tell us
Yeah,
about the science behind it. Um,
definitely.
With that, Jillian, is there anything you'd like to leave us with? Any final parting thoughts of something that you know, for folks to check out, to go follow you? Like, what socials can I plug? What website? Parting thoughts? And any questions for Chad, please ask.
Yeah, I don't really have much to plug. I'm just me. Um, just working in the lab. I mean, I guess the thing, and I know that this audience is, you know, obviously sort of all in on the accessibility train with us, but, you know, sort of doing everything that we can to make sure that everything is as accessible as possible for everyone because it benefits everyone. And so, you know, taking whatever opportunities you have to sort of step up and be like, well, hey, maybe we shouldn't be using the red green color filters on our fish image, on our, you know, in situ hybridization images. You know, like maybe we could change that to like yellow and purple or blue. You know, those sorts of things are small, but they have a big impact on people who need them.
Is there any suggestion that you would have for folks not in the scientific field that they can help make a difference in that? Like, is there a place of advocacy where non-scientists or citizen scientists can help?
I don't know. That's not a. I really don't know. Um, I mean, I think that there are there are accessibility issues in STEM for sure, but there are also accessibility issues in all aspects of the world that we live in. And so I think that I would simply suggest that find something that you're really into or something that you're already a part of and maybe look at it from the perspective of someone who would be hard of hearing and see how their interaction or experience with that would be like.
Yeah, I think
See
that's
if there's a step forward that you can, you know, make there.
nice. I love it. Our silly question for you from Ariella: Do you like Twitch, and do you have any plans to start streaming yourself?
This is my first time on Twitch, so I am new to the community. I don't know that I have any plans to start streaming. Um, maybe if I start doing some pretty heavy microscopy days, I'll pop on and um, you know, just hook a camera up to the scope and take a look at some cool stuff.
You already have your first follower. Uh, we'll give you one of these fancy shout outs. You'll never know if Dr. Freeze might be live, so we'll give her that fancy shout out. Y'all can go drop her a follow. Maybe she'll do some live microscopy.
Yeah.
I know a good, this is Mike's. I know a good stream with microscopy every Monday.
Yeah.
So I guess with that, we have a few announcements. We will be having another Genetic Society of America stream next month in August. Look for the date on that. Dr. Freeze, thank you again for coming on today and spending your time with us.
Of course. Thank you so much.